Hi, I'm Estrella Passerat de la Chapelle, and I'm currently a research associate at the Radiation Biophysics Lab at NASA AMES Research Center. The title of my presentation is Neurovascular Responses to Simulated Deep Space Radiation in a Human Organ on a Chip Model. So I know it sounds kind of complex, but it's basically going to be about the brain and space radiation. Humans have always had a burning desire to explore new environments and to go beyond what is known as possible. So naturally, we want to go to space, but there are limits because this is the most hostile environment humans have ever tried to explore. But we know humans, they're not just gonna stop because it seems impossible. They still want to go. This is why NASA developed the HRP program. Its mission? Protecting astronauts' health and performance from 5 key hazards of spaceflight by researching how astronauts' bodies and minds adapt to the extreme environment of space. And possibly the most critical hazard there is space radiation. But what is space radiation, and why are we saying it is critical? Space radiation is composed of galactic cosmic rays. They're actually a shower of particles that originate in supernovas, are primarily protons with some helium ions and other ions of the elements of the periodic table, all the way through iron. They have major biological effects, and they're very hard to shield from. Both we on Earth and astronauts on the ISS are protected from them by the Earth's magnetic field, but going to the Moon and to Mars will take astronauts beyond that magnetic field, so they will be much more exposed to these particles. So our little astronaut here is not really floating in an empty void, but is rather bombarded with a shower of particles traveling at near relativistic speeds. And the risks are huge. Acute in-flight effects, cancer, cardiovascular disease, and central nervous system risks. The thing is, the brain is the control center for the human body. So if the brain goes kaput, there's a bunch of things that can go wrong, including compromising the whole mission. But studying the effects of space radiation on the human brain is a challenge. How can we study the space brain while here on Earth? Well, research in this field has relied extensively on rodents as well as two-dimensional cell cultures, revealing changes at different levels. But there are some knowledge gaps that cannot be addressed using traditional models, and we need a human, three-dimensional, and multicellular model. Thankfully, there are new technologies emerging to model organs in vitro, namely organs on chips. They are 3D in vitro structures in a microfluidic chip. They're usually formed from commercially acquired primary cells that come from healthy donors. These systems can be exposed to simulated spaceflight stressors or flowness payloads and can provide information in the fundamental biology of responses to spaceflight stressors, of targets for biomarkers and countermeasure development, and comparison between spaceflight and terrestrial diseases. So we decided to use one specific type of organ on a chip, the blood-brain barrier on a chip. Why? And what is the blood-brain barrier? First of all, why? We're interested in the effects of deep space radiation on the central nervous system, and we know that exposing model organisms to simulated galactic cosmic rays or their components leads to behavioral deficits accompanied with neuroinflammation and neurodegeneration at the tissue level. But radiation does not only affect the brain, it also affects the rest of the body. And the brain and the body are connected with each other through the blood-brain barrier. From studying terrestrial diseases, we know that if the barrier is compromised, systemic whole-body responses can exacerbate inflammation in the brain. This is why we're interested in the effects of space radiation on the blood-brain barrier, which are studied very little, especially in human models. But what is the blood-brain barrier? It is a tube of a blood vessel. Its walls are formed of endothelial cells connected with each other through tight junctions, with the brain tissue outside, where particularly important cells are astrocytes. They regulate barrier permeability as well as neuroinflammation and neuronal health. So what exactly is our model? Our blood-brain barrier on a chip is a high-throughput, simplified human blood-brain barrier model. For this, we're using a commercially available system made by Mimetas called the organoplate, which has up to 96 chips per standard 384 well plate. Each chip is composed of two main channels. The top channel is seated within endothelial cells and represents the actual blood vessel. The blood vessel is formed in 3D by the endothelial cells, which in response to the flow of media are forming a tube. Here we can see with just the cell nuclei in blue. The bottom channel, representing the brain side, is seated with astrocytes that are here in green, and they're in a gel which represents the extracellular matrix. So how are we studying space radiation with this model? Well, we seeded the chips with the cells, and 4 days later, we exposed the model to simulated deep space radiation, either to only iron particles as the most biologically active, high-mass, high-energy component of the galactic cosmic rays, or to simplified simulated galactic cosmic rays, which is a cocktail of 5 ions of the GCR spectrum available at the NSRL in Brookhaven. We then quantified the barrier permeability by loading fluorescent dye into the vessel and measuring how much of it leaked into the neighboring brain channel over time. In addition, we investigated radiation-dependent changes in cellular morphology, cell surface markers, and secreted markers of oxidative stress and inflammation. In this talk, I will focus on the acute and subacute responses to radiation and how they're influenced by astrocytes. We observed that during the acute phase, so 24 hours after radiation caused a dose-dependent increase in vascular barrier permeability, which was exacerbated by astrocytes present. Here, the y-axis is the metric of relative fluorescence of the dye that leaked out of the vascular channel. The closer it is to 1, the leakier the barrier. Open bars are without astrocytes, and closed bars are with astrocytes. Blue is no radiation control, and increased shades of red are increased doses of radiation. As you see, radiation and astrocytes both increase permeability, though not always in a dose-dependent manner, which is similar to results reported by other groups in animals. Increased permeability is accompanied by increased astrocytic aquaporin 4 expression. Aquaporin 4 is located on astrocytes and helps them regulate vessel permeability. We also see reduced ZO1 expression. ZO1 is a tight junction protein, and tight junctions normally limit permeability by making the vessel tighter. Here's the actual quantification. So we see an increase in aquaporin 4 and a decrease in ZO1. Now during the subacute phase, so later after radiation, astrocytes begin expressing high levels of GFAP and adopt a reactive, scar-like phenotype. This is similar to astrocyte responses to brain injury or stroke. GFAP expression remains high through 7 days post-irradiation, and interestingly, astrocytes are no longer influencing ZO1 or PKM1 expression or even permeability. But what about the other responses to radiation, such as oxidative stress and inflammation? Again, there's two questions here. What does radiation do to our model? And what is the role of astrocytes in regulating radiation responses? In terms of oxidative stress, radiation increases it and astrocytes reduce it, thus being beneficial. And this both during acute and during the subacute phase. We also saw an increase in inflammatory cytokines and chemokines due to radiation, which was also mitigated by astrocytes. Now, I'll also briefly talk about our current project, the dose-rate effects of ionizing radiation. To achieve chronic exposure to ionizing radiation, we use a system that is very simple. We put a cobalt-57 plate in an acrylic box that we put in an environmental chamber, on which we put the organoplate system. The cobalt plate chronically emits gamma rays at a low dose rate. When we look at the results of the permeability assay, we can see that the blood-brain barrier permeability is increased only after 14 days of chronic low-dose rate gamma radiation compared to high-dose rate gamma radiation, and that antioxidant reduces it. So NACC. We can see that at 7 days, astrocytes are activated only by chronic low-dose rate gamma radiation compared to an acute exposure to the same dose of X-rays. So here. Aquaporin 4, which, if you remember in my previous slide, is expressed by astrocytes to regulate blood-brain barrier permeability, is increased by low-dose rate radiation and by 2 times the same dose of X-rays. Now, in endothelial cells, we can see that PKM1 is increased only by chronic low-dose rate gamma exposure. Finally, we can see that oxidative stress is increased in both low and high-dose rate samples after 7 days, but goes back to normal after 14 days of chronic gamma radiation. This is very interesting and potentially good news, so we'll see you with our new results coming up. So, in summary, we have developed a simplified, high-throughput human neurovascular model and exposed it to simulated galactic cosmic rays or their components, iron ions. We found out that radiation makes the vessels leaky and increases oxidative stress and inflammation for at least a week. In addition, astrocytes have mixed effects early. They reduce oxidative stress, which is good, but also makes the vessels leakier, which is mostly harmful. During the subacute phase, however, astrocytes become reactive, scar-like, and reduce oxidative stress and inflammation. This switch to a protective phenotype during the subacute stage is also seen in neurodegenerative diseases. Now, I believe that these organ models have a very high potential because we can personalize them with cells from a specific subject, say an astronaut. It is small enough to be adapted as payloads to be sent to space, and it can be used to screen potential drugs to eventually find a countermeasure to space radiation-induced cognitive dysfunction. I want to thank my lab, especially Sanaya Verma and Dr. Iglic Ekanavichute, for showing me how to use this model, but also everyone else who helped us and are continuing to help us with this project. And I'm open to questions.